Oops, there we go. If there's no questions, we'll begin. Let me show my screen. Let me remind you that there's going to be a quiz this Wednesday over Lab 5 and Lab 6, Chapter 4 and the end of Chapter 3. Let me also remind you that I got quiz 2A graded, and I'm working on our last lab. So I guess that's Lab 6. And I think I'm only about halfway through. Hopefully I'll get the rest of Lab 6 graded tonight. And I should send out your unknown first test results tonight. Also, somebody can remind me, send out our unknown project tonight, and I'll try and get that done. I should have tried to do that between hand, but I was doing other things for the break between this lecture and the lab. For one thing, I got the lecture up. Usually I don't have time to get the lecture up. Many times I don't even have time for the thing to finish processing on my computer. Any questions about anything? If you're lost on the unknown project, please contact me right away so that you can get up to speed. If you wait to the end of the term, that'll be too late. You're going to need to work on this unknown project. Well, some of you for several weeks. If you're really, really lucky, you can be done in after 1 week. But most people won't be that lucky. All right, so we're discussing Lab 7 today, throat and urine cultures. And you're also working on your unknown project. You should be about done with your infectious disease project. That is due a week from this Saturday. So what a day is that? Probably November 6th. There it is, Saturday, November 6th. You need to turn in your infectious disease papers. Any question about any of that? If not, I'm going to close this. And start talking about the lab. So Lab 7, throat and urine cultures. There is some reading you should do before you do this lab. So please take a look at the reading. There's some video clips in today's labs, but we have not found any good virtual labs. There used to be a virtual lab, but it used Flash. And if you don't know, Flash is no longer working and we haven't found a substitute. So we don't have a virtual lab. Probably between terms, I should go look for virtual labs because I'm going to be teaching online next term also. The learning objectives for this lab, once you finish this lab, you should be able to explain how to properly collect throat and urine specimens for routine culture, be able to list common bacterial pathogens that are isolated from the throat and also from urine cultures. Know the different culture media we use to cultivate these pathogens, be able to recognize which tests are used to identify throat and urine pathogens, and understand the principles and how to interpret the following tests. You should know and understand the catalase test, coagulase, and the oxidase test. We will briefly discuss the XV factor testing, but I'm not going to hold you responsible to knowing that test. Any question about any of that? All right. Once specimens are received in a lab, they are logged and processed in a timely fashion. Specific culture media are used to inoculate and incubate the organisms. Throat and urine specimens use the following media, and these media are used to cultivate common bacterial pathogens isolated from the throat and the urine. So for throat, we really only use two special media, blood agar and chocolate agar. Blood agar is very good and grows many organisms. It is an enriched media, so that if something won't grow on a general purpose media, oftentimes it will grow on blood agar. I'm not saying it always will grow, but it grows more things than regular general media. Streptococcus pyogenes is the most common pathogen that is obtained from the throat, and it will grow on blood agar. And then blood agar also is good because you can detect Streptococcus pyogenes because it has beta hemolytic activity. Chocolate agar is used for Haemophilus influenzae, which is found in children and babies. It's not typically found in older individuals, but Haemophilus influenzae will not grow on blood agar, but it will grow on chocolate agar. Any question about any of that? For urine specimens, we can use blood agar, but they also use blood agar and either McConkey's or EMB, that's eosine methylene blue agar. For blood agar, we can grow the pathogens in urine which are gram positive, like Streptococcus, Enterococcus, and Staphylococcus. However, for the gram negative pathogens, we prefer looking at it under McConkey's or EMB because these two media are selective for gram negative, and they will get rid of most, if not all, gram positive organisms. This is important because many of the urine pathogens, meaning urinary tract infection, are gram negative, especially from the family Enterobacteriaceae. And in that family, E. coli is the most common pathogen in a urine specimen. Another pathogen is Pseudomonas, and it's in its own family. Any question about that? So I guess I should say Pseudomonas aeruginosa. It's in the Pseudomonas family. All right, so let's talk about throat cultures. Sore throats may be caused by viruses or bacteria. If you see white patches on inflamed tonsils, it can be indicative of bacterial infection. If it's just red, the sore throat is red, it's probably a viral infection. This isn't 100%, but clinicians use the looking of the throat to determine whether there's a viral or whether it's bacterial. The most common bacterial pathogen, as I stated, from a sore throat or throat culture is Streptococcus pyogenes. It has a few other names, group A strep or gas for group A strep. I generally call it Streptococcus pyogenes, meaning I name it by its official scientific name. For a strep throat or just even a sore throat, if the clinician wants to test, they will swab. And when you swab the back of the throat and the tonsils. You do not swab the side of the mouth, excuse me, or the tongue. So you should be swabbing where there's redness or white patches in the back of the throat or the tonsils. Patients that test positive for Streptococcus pyogenes are placed on antibiotic therapy. such as penicillin or one of its derivatives. The reason being with Streptococcus pyogenes, the patient generally cannot throw that organism off because of its capsule. It is protected from the host immune system. And so many patients cannot throw a Streptococcus pyogenes infection off. And that's why strep throat used to be so dangerous, because the patient had it for long periods of time, and then it would spread into the blood and then go elsewhere in the body. And the patient just got very sick from it because they couldn't effectively fight it. Nowadays, it's easy to fight off with antibiotics, assuming that it's not antibiotic resistant. Streptococcus pyogenes is fastidious, so it has to be grown on blood agar plate or BAP, and that's simply tryptic soy agar supplemented with 5% sheep's blood. And then you examine the plate for small, clear colonies that have beta hemolysis. Let me see if I go right here, showing it, see if I can blow this up. I don't know how visible that is, but in the center there, that's the colony. And then this white ring around it is beta hemolytic activity in the blood agar plate. This is not beta hemolytic activity. In fact, these don't have any hemolysis. In this region where it's darkening, there is alpha hemolytic activity. So a normal throat, and then hemolytic colonies from the throat culture. All right, just looking at this, you can't be certain that this is beta hemolytic activity, that it's strep throat. And so you need to do further testing. If the throat culture shows beta hemolytic activity, you then need to run 2 biochemical tests to prove that it is Streptococcus pyogenes. You run the catalase test and the basotracin susceptibility test. Catalase test is a test where the enzyme produces bubbles from hydrogen peroxide. So you put hydrogen peroxide on the slide, shown here, and then you add the bacteria. If you get bubbles, then the organism is catalase positive. If there's no bubbles, then the organism is catalase negative. Streptococcus pyogenes is catalase negative. Most organisms that grow in the air are catalase positive, because most organisms that grow in the air are exposed to oxygen, and oxygen can produce hydrogen peroxide, and this is a toxic substance to cells. And so they need to remove the toxic substance. And most cells that live in the air have catalase, like your skin, and it can convert hydrogen peroxide into water and oxygen. The making of oxygen produces the bubbles. Any question about any of that? All right, there are 2 families though that do grow in the air, which are catalase negative. And that includes the Streptococcus family in the Enterococcus family. They're both catalase negative. So if it's catalase negative, then you run the bacitracin susceptibility test. Bacitracin is an antibiotic, and Streptococcus pyogenes is group A strep. It's sensitive to bacitracin. So here's a disc that we take the bacteria that was grown here and then spread it everywhere, well, I should say in this region of the disc. And generally they grow it on the blood agar plate. And then you put the paper disc containing the bacitracin down. And if you have a significant zone of inhibition around the paper disc, then this is susceptible to bacitracin, and Streptococcus pyogenes is susceptible to bacitracin. The other beta hemolytic bacteria that are catalase negative are not susceptible to bacitracin, and they generally have no zone of inhibition. Okay, you do need to measure that zone of inhibition to make sure that the organism is resistant to bacitracin. Is that right? No, sensitive to bacitracin. This is resistant to bacitracin. All right, any question about that? You measure then, if the diameter is a certain size, and you compare it to the chart of the standards, and if it's above that size, then we say it is sensitive to bacitracin. Streptococcus pyogenes is sensitive to bacitracin. Another organism that is clinically significant and isolated from the throat is Haemophilus influenzae, which is a gram-negative coccobacillus. It is one of the most common causes of ear, eye, and sinus infections in babies and young children. It's not common in adults and even older children. It does not grow on blood agar, so you need to grow it on chocolate agar. Chocolate agar is the same as blood. It's just that you add the blood when the agar is still very hot, and that lyses the blood. Haemophilus influenzae does not lyse the blood to get the nutrients it needs from red blood cells. But if you provide it with chocolate agar, you can get growth of Haemophilus influenzae on the plate. This is showing you that this plate has been streaked with Haemophilus influenzae right here. And as you can see, there is no growth in this region of the plate. And the streaking goes all the way down to here. And you can see Haemophilus influenzae growing in this region of the plate. The reason is this right here is a streak of Staphylococcus aureus. Staphylococcus aureus has beta hemolytic activity, which you can't really see it in this plate here. And because it lyses the red blood cells, that will release the nutrients from the red blood. And then Haemophilus influenzae, which won't grow here, if you streak it over here, it will grow in this region of the plate because of the Staphylococcus aureus. Any question about any of that? There's actually a test for that. I think we will run into it later in the lab today. But maybe it's in a future lab, I don't remember. A Haemophilus influenzae is a grayish, transparent colony with an irregular border. You have to blow that up to see that. It doesn't grow on blood agar. unless it's right next to the Staphylococcus aureus. And usually you don't grow two things on a plate, but it will grow on chocolate agar. All right, any question about the throat cultures? If not, let's turn to the urine cultures. Urine cultures are used to diagnose urinary tract infections. Bacteria can travel from the lower urethra, where it is normal, up to the upper urethra, and then up into the bladder. If it goes into the bladder, it can cause a urinary tract infection, and females are more disposed to get a urinary tract infection than males because of their shorter urethra. There are 3 ways to collect a urinary specimen. Patients prefer the clean catch midstream, which involves urinating and only collecting the urine in the middle of the urine stream. You clean the urethra beforehand, and then you urinate, and then you collect it midstream, and that's called clean catch midstream. Patients much prefer that than sticking a catheter up the patient and then getting the urine specimen. And then the 3rd way of getting a urine specimen is suprapubic aspiration, where they literally poke a needle into the bladder and then sample the urine from the bladder directly. This suprapubic aspiration is a very clean way of getting the urine, and you're unlikely to contaminate the urine with any bacteria if you do it that way. The catheter is much cleaner, but obviously as you're putting the catheter up the urethra, it can pick up bacteria and then put the bacteria into the bladder. Still, the catheter is much cleaner, has much less contamination of bacteria than the clean catch midstream method of getting urine. Any question about any of that? Once you get the urine specimen, 1/1000th of a millimeter, that's 0.001 milliliter of urine is placed onto a non-selective media like blood agar using a special inoculation loop, where the loop itself has only 1/1000th of a milliliter. And if you can see that, let me blow that up a little. There's the loop there. And this is trying to show you how small that loop is. And you get the urine in there and then streak it over the blood agar plate. Usually you go like that and then like that. And then get it to grow. You should also streak it on a McConkey's or EMB plate so that you can see gram-negative organisms, which could be the pathogen. And then on the blood agar plate to get both gram-negatives and gram-positives growing. Any question about that? So for a urine specimen, it should both grow on blood agar and either on McConkey's or EMB. We grow it on McConkey's or EMB to look for gram-negatives, which are the most common urinary tract infective organisms. And the very #1 most infective organism is E. coli. All right, any question about any of that? Interpretation of a positive urine culture is multifaceted. There are a number of factors that need to be considered for your interpretation. The type of collection really will depend on your interpretation. Avoided versus catheterized method or suprapubic aspiration. The gender of the patient will determine the interpretation. Females have a lot more bacteria in the clean catch method of collecting urine than males. The number of colonies per mil of urine will be used to make your interpretation. And then the presence of polymicrobial growth. If there's multiple organisms isolated, it may mean that there was a poor collection method. Males generally have 50,000 colonies per mil in urine. I assume this is with a clean catch method. And females generally have 100,000 or more colonies per mil in urine. All right, a couple of tests are used once you get bacteria growing that are isolated from the urine. Remember, if you use the clean catch method, you're going to get a lot of normal bacteria growing. So you need to run the test. One of the tests that's used is the enzyme coagulase. Coagulase is used to differentiate Staphylococcus aureus, a human pathogen, from other Staphylococcus species. Staphylococcus aureus is coagulase positive. The other members of the Staphylococcus family are coagulase negative. Coagulase contributes to the pathogenicity of Staphylococcus aureus by converting blood plasma, the fibrinogen in blood plasma, to fibrin, which then results in a blood clot. So fibrinogen or blood plasma plus coagulase equals a clot. And Staphylococcus aureus has the enzyme coagulase. There are two tests. You can do in the slide test, which you would get clumping showing like this. A negative coagulase test is like that. You can also do the serum test in a test tube. And this is the normal tube with serum. And then you put the bacteria in. And if it's coagulase negative, the serum will be liquid. And if it's coagulase positive, it will clump. And so the serum will clump like that as opposed to being like that. Staphylococcus aureus, like I said, is coagulase positive. The most common bacteria isolated from a urinary tract infection is E. coli, which is in the family Enterobacteriaceae. To see E. coli, we grow the bacteria on either McConkey's media or EMB. There is another species, though, which is not in the family Enterobacteriaceae, but it is a gram-negative organism, and that's Pseudomonas aeruginosa. So it's gram-negative, so you'll see it on the McConkey's or EMB plate, but it's different than the Enterobacteriaceae because it's aerobic and it's non-glucose fermenting. All of the members of the family Enterobacteriaceae are facultative anaerobes, and they ferment the sugar glucose. So the oxidase test is used to differentiate the Pseudomonas from the family Enterobacteriaceae. The oxidase test has reagents in it and it'll turn purple in the presence of oxidase. So this is oxidase positive. And this region over here would be oxidase negative. It's not purple. What we're doing is we're looking for the cytochrome C. And cytochrome C oxidase will turn the oxidase test purple. All right, any questions about urinary specimens? So in lab 7, you're not physically going to be performing any of these activities. However, you will be responsible for knowing how to perform them. You can read about them, and we have video clips on the different procedures and tests. Like here's a video clip on running the catalase test. There's a video clip on running the coagulase test. And there's a video clip on running the oxidase test. You do not need, you cannot do the virtual labs because they run flash. And I'm not going to try and explain to you how you could get that to run. All right, so once you've finished reading through the lab and watching the video clips, get the laboratory exercises for lab 7, and then answer the questions. Many of the questions are testing you on the different tests and how you use them. Let's see how many tests there are. There's 13 tests, 13 questions. All right, any questions?
Do we not have to answer 8C since it's crossed out?
That is correct. I left it in there, but I crossed it out because it's not discussed in the lab. You'd have to do research to find it. And I just said, you know, it's not important. I'm crossing it out. Okay. You don't need to do 8C. Good question. All right. I will be here until 8 o'clock. It's now 7:05 to answer any questions. But don't wait till the end because when the last student logs out, I'm going to log out as well. All right. So work on the lab.